Hello to our friends joining us via recording. We are reviewing once again for the final exam. To help us get ready for that exam, we're going to focus tonight a lot on the Crossbridge cycle, and we're going to talk about the things that are inside the cell, the ions and the macromolecules inside the cell. So before we started this recording, we were working with our groups to figure out which particular ions or macromolecules that are described by each of these statements. So we're going to go through one by one and see if we can fill in these blanks together. The first statement that we had to come up with was figuring out which ions help us to polarize the membrane. Quick review, and I know we already talked about this here in class, so let's just make sure we really remember. When we talk about an ion, once again, what does that word ion mean? When we talk about ions, let's get a color here. Yeah, ions are something that has a charge, anything that has a charge. So I'm going to underline all my ions here. Everything that is an ion that we've listed. When we talk about ions, we are talking about what I use to polarize the membrane. In particular, this word polarize, this was a Unit 1 word. When we said that the cell membrane was polarized, that meant that there was a different charge on the inside compared to the outside. Now, I'm going to make you stretch your minds a little bit here, so don't freak out too much. I know we know this. We talked about a special kind of transport that in particular we said is what we used to polarize the plasma membrane. It was called primary active transport. Primary active transport. Yeah, Miriam's totally right. Our example of primary active transport was what we called the sodium-potassium pump. The sodium-potassium pump. The job of the sodium-potassium pump is to pump sodium to the outside and to pump potassium to the inside. But because we don't move the same number each direction, we end up with a polarized membrane, with a membrane that has a different charge on the outside compared to the inside. So when I'm talking about the particular ions that help me to polarize the membrane, the particular ions that polarize my membrane are the two that are involved in the sodium-potassium pump. That's sodium, and that's potassium. The two main ions that polarize the membrane, that make us different inside versus outside, these are the main ones that help us to polarize the membrane. We could also toss on there, just for our remembrance, especially because we're doing that salty banana, right? The other one that helps out with this is chloride as well. So sodium, potassium, and chloride, these are the main ions, but especially these two, that help us to polarize the membrane, make it different inside compared to outside. My next statement asks us about a macromolecule. When we talk about macromolecules, these are things that are big. Ions are really small. Macromolecules are much bigger. So we have two macromolecules that are listed here. We have the macromolecule of glucose and the macromolecule of proteins. When I talk about the type of macromolecule that helps us to do chemical reactions, which type helps us to do chemical reactions? Yeah, so proteins are what help us to do chemical reactions. Remember that we had a special name for the kind of proteins that do chemical reactions. That special name that we called them was enzymes. Yeah, exactly, enzymes. So when we talk about one of the functions of proteins in a cell, its job is to act like an enzyme, or its job is to make chemical reactions happen more quickly. If we didn't have any enzymes in the body, you probably wouldn't survive because it would take way too long for your body to do things. So one of my functions of proteins is to act like enzymes, to make chemical reactions happen. We've got another macromolecule question down here. Are proteins a cell's favorite food? Are proteins what cells like to eat? Yeah, everyone's saying in the chat the correct answer. Proteins are not their favorite food, right? Their favorite food is glucose. That's a cell's favorite food. So remember when we were talking about active transport, that a cell will spend its energy monies to buy more glucose or to bring glucose inside, even though there's too much or there's a lot of glucose inside already. We spend energy in secondary active transport to bring glucose inside because that's our favorite food. We're going to bring that in. So the function of glucose, which is an example I'll mention for us in parentheses here, of a carbohydrate. A carbohydrate. Glucose is the macromolecule that cells use to make ATP, use to make energy. Let's switch over really fast while we're on the macromolecule train over here to our types of macromolecules that help cells to communicate. What did we think about the type of macromolecule that helps cells to communicate? Any guesses? Yeah, so a couple of us are chiming in, and we're chiming in correct. This is another job of proteins. When we talk about the way that cells communicate with one another, we are talking about chemically gated channels, specifically chemically gated channel proteins that open up with the type of chemical called neurotransmitters. Neurotransmitters. So another job of proteins is to help cells to communicate with each other. One cell spits out a chemical, a neurotransmitter, that comes down and attaches to a protein on the plasma membrane of its neighbor, allowing that cell to communicate or to process what the neighbor is saying. Okay, here's one, we got another macromolecule question up here too. When we talk about the macromolecule that gives a cell its shape, who's going to give us our shape? Yep, again, we got proteins. Proteins that help us to give our shape. When we talk about the shape, we have a special name for my proteins that give the cell its shape. Does anyone remember what all those proteins are called? Yeah, there we go. Miriam already chimed it in. That's our cytoskeleton. Cytoskeleton, that's made out of proteins. So the shape of the cell is because of proteins. So check this out. When we're thinking about what proteins do in a cell, they act like enzymes, which makes chemical reactions happen. They give a cell its shape when they form the cytoskeleton. They help cells to communicate with each other. The one other big thing that we could add to our list here are a couple other big things. Let's mention that proteins also help us to transport things across the membrane. Another one of their functions, transporting things across the membrane. And we could also say that another function of proteins is to attach cells together. Big picture idea when you're thinking about the macromolecules, almost everything that a cell does, its big picture functions, is probably done by proteins. Proteins are very versatile. We have a whole lot of them. They do a lot of different things for us. One of the proteins we talked about this semester was troponin. When we talked about troponin, what was this protein involved in? Where did we talk about troponin? Yeah, we talked about troponin when we were talking about the process of muscle contraction. Troponin is one of my proteins. Yeah, it's found inside the thing called the sarcomere. Troponin is one of my regulatory proteins, just like Miriam said. It's a regulator. The way that troponin can regulate muscle contraction is because it's sensitive to an ion. One of our ions from the list up there comes down and changes its shape when it attaches to troponin. Which of those ions can attach to troponin? Who's able to do that? Yeah, calcium. Calcium is my ion that attaches to troponin. Remember that troponin is like a pushpin. If calcium attaches to my pushpin protein, my pushpin pops out. That causes my spaghetti noodle, called tropomyosin, to also change its shape. That's going to allow muscle contraction to happen. So when we talk about the ion that helps us basically start muscle contraction, it's calcium. Calcium gets rid of that regulatory protein, allows us to do the process of contraction. Hey, for us to do contraction, we release calcium from the sarcoplasmic reticulum in a muscle cell. And the way that I release that calcium from the sarcoplasmic reticulum, the way I know that I need to, is because I have what's called an action potential. An action potential. When we talked about action potentials, we talked about them in the context of Dr. Ollis's favorite graph. An action potential. It's a change in the membrane charge, and it moves all the way across the membrane. So when we talk about the ions, and I apologize, we were missing a word here, these ions are used to generate an action potential. When we talk about the ions that do an action potential, I'm talking about who comes in on this side and who goes out on this side. Which two ions help me to make an action potential? Which ions do I use on that graph? Yeah, exactly. I use sodium to rush in. It depolarizes the membrane. And I use potassium, which rushes out, and it repolarizes the membrane. These are my two ions that I use to make my membrane charge positive and then make it negative again. I use that action potential to go down to my sarcoplasmic reticulum and cause it to spit out calcium. So, action potential caused by sodium and potassium. An action potential, that change in membrane charge, is triggered when a neurotransmitter attaches to a muscle cell. But when we're talking about the neuron that spit out that neurotransmitter, there is a particular ion that when it comes into a neuron, it causes the neurotransmitters to peace out. This ion comes in, neurotransmitters leave. Who comes in and causes neurotransmitters to leave? Yeah, several of us chiming in the chat. Calcium comes into the axon terminal of a neuron. When calcium comes inside, neurotransmitters spit out. They can't stay together. Okay, big picture. When we're talking about what things do in cells, proteins do a ton of stuff, right? They act like enzymes. They help us to form the cytoskeleton. They help us to communicate with our neighbors or attach to our neighbors. They help us to move stuff across the membrane. When we talk about carbohydrates, or in particular glucose, really the only thing that glucose is there for is our food source. I mean, that's an important job. So its only job to feed the cell. When we talk about ions, ions make our membrane have a different charge inside versus outside. Ions help us to change the membrane charge all across it. They help us to spit out neurotransmitters. They regulate the process of muscle contraction. Ions do a lot of different things in the cell as well. What questions do we still have about ions and macromolecules? Go for it, Miriam.
So one thing we've talked about in the group work, what confuses me is that the question which says the macromolecule, which allows the cell to communicate with its neighbors. Well, me personally thought it's calcium. From that graph, that's the only ion that will allow the cell to communicate with others, sending messages out. But you said the keyword is the macromolecule versus ion. So if we substituted this word with ion, would that be a correct answer? Is that calcium will allow cell to communicate with neighbors?
Yeah, we could definitely do that. So we kind of said that in a different way.
Right.
Here, so let's reword that the way that you mentioned it, because you're totally right. This ion allows neurons to communicate. How did we? Yeah, communicate with their neighbors. Type that. Perfect. All right, let me move it. Yeah. So either of those statements would be correct about calcium. But it is, like you mentioned, it's that key word. We mentioned here that we're talking about an ion. If we're talking about a macromolecule, we need something bigger. So we're looking for what the ions that rush in that cause the neurotransmitters to come out, we want to know what they'd be attaching to, and they'd be attaching to a protein.
Okay. One last clarification on the other question and about on the top of this one. The ions that generating the action potential, I always had it in my mind as the very first ion that caused the action potential to happen, which is the one like pulls the trigger. Why we are counting potassium as one of it's like not generating the action potential, like the way it happens that the potassium is fixing or getting the cell back to where it's supposed to be. So it's not generating the action potential, or I misunderstood that from the beginning.
Yeah, so when we talk about an action potential, the key to remember with that is it's a charge change that moves all across the plasma membrane of a neuron or of a muscle cell. So if you look in your notes, it's going to be in lesson number 9 and in number 10. There's that picture that has the 3 tubes that represent the axon of a neuron, or that represent the sarcolemma of a muscle cell. And it shows how first sodium rushes in on the very left side of the picture. And then if you go to the line below it, potassium comes in right after it. Meanwhile, the next part of the membrane, sodium's rushing inside. And then potassium comes in right after it. So in an action potential, we change our membrane charge and it moves all across the membrane. The whole process of going from low to high to back to normal, this whole thing is an action potential. So an action potential isn't just depolarizing, it's also repolarizing. Both of those are parts of an action potential. Does that clarify it? Maybe Miriam's typing for me. Other Miriam, did you have a question you wanted to ask as well?
Yes, Professor Hollis. For troponin and tropomyosin, at first I thought it was tropomyosin that needs the calcium ion to change its shape, especially also when we're talking about for the process regarding muscle contraction. I thought it was the tropomyosin that uses the change its shape to be able to do muscle contraction. And then I got troponin that just uses a different ion other than that to change its original shape as well.
Yes, so this would be a good time here. We'll switch over to talk about our statements here, because we mentioned the protein troponin, and we also mentioned the protein tropomyosin. When we're talking about muscle contraction, think about troponin like a pushpin protein. This pushpin protein is holding the other protein called tropomyosin in place. If we were looking at a picture of the actin protein, the one that has the attachment sites for myosin, what covers up the attachment sites for myosin is the protein called tropomyosin. So tropomyosin, I like to call it the wet spaghetti noodle. It's the one that's sitting directly on top of the places where myosin would attach to actin. It actually blocks the sites. But like a wet spaghetti noodle, it's not going to stay in place unless we have something that will hold it there. So what holds tropomyosin in place is my pushpin protein called troponin. Troponin is the only one that has a place for calcium to attach to it. Tropomyosin doesn't care about calcium. What tropomyosin cares about is whether or not troponin is pushing on it and keeping it in place. When calcium attaches to troponin, it can't keep pushing on tropomyosin. So when calcium attaches to troponin, the pushpin protein pops out, and I don't have anything to push tropomyosin back into place. So short answer to your question is calcium does attach to troponin. Troponin attaches to tropomyosin. And tropomyosin attaches to actin.
Okay, but there isn't an ion that specifically attaches to tropomyosin during the muscle contraction process. Just to troponin, it's calcium, and troponin attaches to tropomyosin, and then tropomyosin attaches to actin, correct?
Correct. Yep. So tropomyosin does not interact with any ions in the process of muscle contraction. That's correct.
Okay, gotcha. That was throwing me off. Thank you.
Yeah, you're welcome. Okay, so let's look at our statements. We have 3 statements here that we needed to fix. The first statement says that the thick filament has actin, myosin, and troponin. Let's assume that we want to talk about the thick filament. Let's assume we're going to keep this talking about the thick filament. If I'm talking about the thick filament, what protein or proteins do I find in the thick filament? Who lives in the thick filament? Underline in red. Yeah, the thick filament only has myosin. The only protein in the thick filament is myosin. Everybody else that we have listed here, actin, troponin, tropomyosin, all of those are part of what? What is actin, troponin, and tropomyosin? What are those all part of? Yeah, those are part of the thin filament. The thin filament. The main protein that makes up the thin filament is actin. And then remember, we've got these 2 regulatory proteins that are on top of it. So when we talk about the thin filament and the thick filament in the sarcomere, the thick filament only made of the protein myosin. The thin filament made of actin, troponin, and tropomyosin. So through 2 ways you could have fixed this statement. You could either tell me about the thick filament or the thin filament. Doesn't matter, either way. Let's go to our second statement here. Our second statement here talks about ATP attaching to myosin in a step called Crossbridge Formation. There are 2 ways that we can fix this statement. First, let's assume that this part is correct. If we're talking about ATP attaching to myosin, which stage of the Crossbridge cycle does ATP attach to myosin? ATP attaches to myosin. Yeah, Gloria's right. In the stage called Crossbridge Detachment. Crossbridge Detachment. If you remember, when we were talking about the thing called rigor mortis, when your muscles get really stiff after you die, this is the problem. You stop making ATP. ATP is what attaches to myosin and allows myosin and actin to let go of each other to keep your muscles from getting stiff. So ATP attaching to myosin, that happens in the stage called cross-bridge detachment. Okay, that's the first way I could fix this statement. The second way I could fix this statement is assuming that I want to know what happens in cross-bridge formation. In cross-bridge formation, what does myosin attach to in cross-bridge formation? Yeah, in crossbridge formation, we form the crossbridge where actin and myosin attach to each other. So if we wanted to do crossbridge formation, remember a crossbridge is just actin and myosin touching, holding on to each other. Actin and myosin attach to each other in crossbridge formation. Hey, then we move into the stage called the power stroke. Now, the power stroke is not when myosin breaks down ATP. If I wanted to change this statement and I wanted to talk about what happens in the power stroke, what are some of the things that we came up with or that we might think of now that happened during the stage called the power stroke? What happens during the power stroke? Oh, green's a bad pick. Let's do orange. The power stroke. Perfect. Yep. So in the power stroke, this is when movement actually happens. So actin slides past myosin. The reason that actin can slide past myosin is because myosin lets go of ADP. If it lets go of that, it allows actin to slide past. So when we talk about the power stroke, this is not when myosin breaks down ATP to form ADP and inorganic phosphate. This is not when I chop up my energy source. In the power stroke, this is where I generate the power of movement. So when I'm pulling things past each other, I do it because I spit out ADP. There is a step, though. It's the one step that we have not talked about in the Crossbridge cycle, the step where myosin does actually break down ATP. What is the name of the step where myosin breaks down ATP? What did we call that step? Switch colors here. Yeah, so the step where myosin actually breaks down ATP, where it steals the energy of ATP, this is called cocking the myosin head. Oops, myosin head, or sometimes one of our videos calls it reactivation of the myosin head. means the same thing. I like reactivation because it gives you an idea that this is where I get energy back into myosin. So when myosin is holding on to ATP, when it chops up ATP into two pieces, myosin gets that energy, and myosin goes from being in a bent over position into an upright position, or what's called the cocked position. This is where it's ready to attach to actin again. So we've just described in words the 4 stages of the Crossbridge cycle. We're going to look at them in pictures, but before we do, are there any last-minute questions about this slide before this slide goes away? Yeah, I was going to say, or give me an emoji. How are we feeling? What are we thinking? Yeah, working on memorizing. Yep, absolutely. All right, well, let's take these words that we just talked about and let's pair them with the pictures that you have in your notes packet. Can somebody help me out? I know that we are in lesson number 9. What page are we on in lesson number 9? Okay, so we're spanning kind of across pages 6 and 7. Excellent. Okay. So this is when we first start talking about the Crossbridge cycle, and it has labeled for us the 4 stages of the Crossbridge cycle. Remember that we call it a cycle because it keeps going and going and going. So here's the process. Let's talk through it together and use our pictures to help us. Before we even go into the Crossbridge cycle, notice right here on my picture how it shows that there's calcium. So before I can even start the Crossbridge cycle, I have to spit out calcium from the sarcoplasmic reticulum. Because remember, that calcium has to go down and attach to troponin. Once it attaches to troponin, tropomyosin, the spaghetti noodle here, slides out of the way. If we are not doing the process of muscle contraction, if there's no calcium, remember that this spaghetti noodle here, it would be on top of all of these little spots. The little spots are the places where myosin can attach to actin. If we don't have calcium, that's where myosin lives. It lives on top, or tropomyosin lives on top of those places. So we've spit out calcium. Troponin, my push pin popped out. Tropomyosin moved. I've got a space for myosin to attach to actin. So step 1, crossbridge formation. In the step of crossbridge formation, I form an attachment between myosin and actin. This is always going to happen as long as the actin, the myosin binding sites on actin are available. So as long as myosin is in the upright or the cocked position, and as long as there's an open space on actin, I'll always form a crossbridge. So myosin attaches to actin. That's the crossbridge formation step. Second step, called the power stroke. The power stroke is where we actually see movement. So see my little arrows? We actually see movement. What causes us to have movement is now that myosin is attached to actin, now that we've formed this attachment, myosin says, I don't need these things anymore. So myosin spits out ADP and inorganic phosphate. We can see them leaving here. When myosin spits them out, the shape of myosin changes. So notice how it used to be kind of laid back with its head upright. When I get rid of ADP and inorganic phosphate, myosin ends up more upright and its heads are bent over. That's what causes actin to move. So in the power stroke, we spit out ADP, an inorganic phosphate, and actin slides. That's what happens in the power stroke. At the end of the power stroke, actin and myosin are attached to each other, and myosin has no energy, and we're stuck at the end of the power stroke. That's why we do our next step called crossbridge detachment. In crossbridge detachment, we're detaching myosin from actin. We're separating these two proteins from each other. The way that I separate them from each other is I give myosin something that's shinier than actin. Myosin says, ooh, ATP, I like that. When myosin grabs the ATP, it lets go of the actin. That's crossbridge detachment. When myosin has the ATP, it's no longer attached to actin, but see how it's still kind of laying down? It's in a low energy position. The last step of the crossbridge cycle is cocking the myosin head or reactivating it. It's taking the myosin head from the bent over, the slouching position to the upright position. It takes energy to make myosin stand upright. So what I do is I hydrolyze ATP, or I chop it up. I get rid of some of its energy. That energy goes to the myosin head to make it stand upright so that it's ready. If there are actin sites on actin available for it, it can go through and form that attachment to do the crossbridge cycle again. 4 stages to the process of the crossbridge cycle. We attach myosin and actin. We slide myosin and actin away or toward the middle of a sarcomere. We make them let go of each other, and then we change the shape on myosin so it can do the whole process again. What questions can I answer at this point? Miriam, no, I believe there are not these pictures on the exam. So we want to make sure we can describe what's happening in these stages. You'll probably just get the name of the stage. Yeah, so Christina asked the question, before any of this can happen, we need to have calcium released, and that is correct. Yes, because if I haven't spit out calcium, all of my myosin binding sites that we see right here, all of them would still be covered up by tropomyosin. So it wouldn't matter if myosin was in the cocked position. If it has no myosin binding sites on actin to attach to, then we can't do the crossbridge cycle anyway. So typically what's happening in your body when you're not doing muscle contraction is all of your myosin heads are chilling out in the cocked position. They're waiting to do muscle contraction. When we first release that calcium, then we'll jump right into the process of crossbridge formation, and we'll cycle through this over and over again. So when you're waiting for muscle contraction to happen, you chill out right here, and we spit out the calcium, and the whole process begins in earnest. Another question, where does the ATP come from? So if you remember, when we were talking about the types of skeletal muscle tissue, we said that we have some skeletal muscle tissue that is glycolytic and some that is oxidative. Remember that those were the 2 ways that a muscle fiber can make ATP. So the short answer to where this came from is the cell made it, either in its mitochondria or using that process called glycolysis. So this was floating around inside the cell, and when it bumps into myosin, it causes detachment to happen. Let me see. So Andrea, to your question about ADP and phosphate, it's not that they reattach to myosin. So here, let's talk a little chemistry here. And I'm going to write. So hopefully my writing isn't too terrible. When we talk about ATP, we've got our adenosine, and then it's called triphosphate. So phosphate is PO4. Adenosine triphosphate. I really should have typed this. Adenosine triphosphate, ATP. I've got 1, 2, 3 phosphate groups that are all attached to adenosine. So this molecule right here, ATP, adenosine triphosphate, 1, 2, and 3 phosphate groups. What happens between the detachment stage and the cocking the myosin head stage is I chop up this chemical bond right here. So this whole molecule, this was one thing, this was ATP. ATP turns into ADP and phosphate because basically, here, let me draw a little myosin here. So this whole thing is attached to my myosin. So here's my myosin golf club head here. This whole thing is attached to myosin. What happens in the cocking of the myosin head stage or the reactivation stage is I just break this chemical bond right here. So now I've got ADP with 2 phosphates, and I've got this phosphate group by itself. So I just took the thing that I had right here, and I turned it into something new. All I did is I chopped up that phosphate or that bond. I broke that bond. That allows myosin to stand up to be ready. I'll spit it out when I get over here, but this is going to be a new ADP and phosphate, a new one that came when I chopped up ATP. Let me see. Yeah, so the question is when we get through all 4 of these steps, then it just starts over. That is correct. Yeah, for as long as your cell has ATP, so as long as it has energy, and as long as we have calcium released, we will just keep going and going. So we'll form a cross bridge, we'll pull on actin, we'll let go of myosin and actin, and get myosin in the right shape. And if myosin can still attach to actin, we'll do it again. We'll pull on it, we'll let go, we'll get back in the right shape. Yeah, this cycle will just continue forever and ever as long as there's ATP and as long as there's calcium. If I have those things, I will just keep doing this. For the question, can there be problems with this process? We talked a little bit about some of those problems. Problems, for example, like what causes rigor mortis. If I don't have ATP, then my muscles get stuck together, and that would be a problem with this process. The other thing that could be problematic with this process is if I don't have any calcium. If I don't have any calcium, I can't do any of this stuff because I can never do the Crossbridge Formation step. So we could have problems with this process if we messed up the things that are involved in it. What other questions do we have about the Crossbridge cycle? Okay. Tyranny asked where the calcium came from. So when you talk about the calcium at the neuromuscular junction, that particular calcium is going inside the neuron. It's not going over to the muscle cell. The calcium that we're talking about here is the calcium that we spit out from the sarcoplasmic reticulum. So the stuff that we had stored inside the muscle cell, we spit that out into the sarcoplasm, the soup inside the cell. But that's a different pool of calcium than what we talked about with the neuron. Yeah, but it is calcium for both of them, so that's a good connection to make. All right, so I'm looking at my notes here. Let me put the question to the class. We've got about 20 more minutes. What do we want to talk about? Gloria's voting the types of skeletal muscle. What other votes do we got? We could talk about the types of skeletal muscle. We could talk about prions. We could talk about protein shape. If I have no other votes, I'll talk about skeletal muscle. Good luck, Christina. Okay, well, Miriam, why don't you save your integument question for the end? I will definitely take it. We did talk about the integument a little bit this morning, so we'll save that for the end. What we'll go ahead and do now is we'll talk about the types of muscle tissue, what we could use them for, or what they're good at. To help us do that, we're going to build a table together. So what I think you should do is grab a piece of paper or something to take some notes on. We're going to build a compare and contrast table together to help us out with our types of muscle tissue. So when we talk about skeletal muscle tissue, we have three types. We have what's called fast glycolytic muscle. We have what's called fast oxidative muscle. And then we have what's called slow oxidative muscle. With these types of muscle tissue, some of the things that we need to know how they make ATP, so where they get their energy from. We need to know if they do a strong or weak contraction. We need to know how fast they run out of ATP. We should also know, do they need oxygen? Do they need O2? Here's some of the major things that we need to make sure we know about these types of muscle tissue. So let's start with this first part of them, how they make their ATP. When you look at the types of muscle tissue, their name tells you how they make their ATP. So particularly the words that are going to tell us how they make their ATP are the words glycolytic or oxidative. Glycolytic or oxidative. When I use the word glycolytic, do we remember what glycolytic meant? What does glycolytic mean? Anybody remember? Yeah, so glycolytic looks like glucose, right? So when we talk about a glycolytic fiber, they're using a process called glycolysis to make their energy. So we're breaking down stored sugar. A glycolytic fiber is something that makes their ATP by breaking down sugar that they have stored inside of them. When you talk about glycolytic fibers, because they have sugar stored already, because it's inside, this kind of muscle fiber can make ATP really fast because we can break down sugar really easily, really quickly. So fast glycolytic muscle breaks down stored sugar. Glycolytic means we use glycolysis. We're using that stored sugar. When we talk about oxidative, does anyone remember where ATP came from if we have an oxidative type of muscle tissue? Oxidative? Yeah, excellent. So Miriam's mentioning the particular organelle that I use to make ATP in oxidative fibers. I use the mitochondria to make my ATP? The mitochondria has something inside it called the electron transport chain. The electron transport chain, or abbreviated ETC. The way that you can remember the electron transport chain with this word oxidative is because at the end of the electron transport chain, I have to have oxygen. For the electron transport chain to work, I've got to have oxygen. So if you see this word oxidative, oxidative means I use the electron transport chain to make my energy. If you see the word glycolytic, that means I use glycolysis to make my energy. Two different processes to make my energy. The way we know how we're making it is either glycolytic or oxidative. The next thing the name of these muscle cells tells us is whether we're making a strong contraction or a weak contraction. That information comes from the first word in their name, whether it's fast or whether it's slow. When we talk about a fast muscle, what type, we're talking about a type of protein that can break up ATP really quickly. So when you're thinking about a lumberjack, for example, that's chopping up wood, if you've got a fast lumberjack, they can chop up wood really quickly. If you throw all of that wood into the fire, you make a really strong or really hot fire really quickly. So when you see this word fast in the names of these types of muscle tissue, what that means for us is that we've got a strong muscle contraction. A strong muscle contraction because it's a fast oxidative fiber. When we talk about a slow oxidative fiber, the kind of contraction that a slow fiber is going to make is going to be a little bit weaker because the rate that I chop up my ATP is a little bit slower. I don't make as much energy or as quickly. I don't make my energy as quickly because I'm not chopping it up. So strong contractions come from types of muscle tissue with the word fast in their name. Slow muscle fibers are going to make a weaker contraction. But here's the thing with my slow oxidative fibers, or with slow in general here, if I chop up my ATP more slowly, I don't run out of ATP very quickly either. So when we think about how quickly we run out of ATP, slow oxidative never runs out of ATP. We never run out of it because I'm chopping it up slowly. That gives me a weak contraction, but I chop my ATP up slowly. And when we talk about the electron transport chain, that makes a ton of ATP. So if you have an oxidative fiber, it makes a lot of ATP. You don't run out of ATP as quickly if you're making a bunch of it. So when we talk about slow oxidative fibers, we would say that these fatigue slowly. Fatigue means I ran out of ATP. I'm going to fatigue slowly if I chop up my ATP slowly and I make a whole bunch of it. When we talk about fast oxidative fibers, oxidative still means that I make a lot of ATP in my mitochondria. But remember that I have this word fast, which means I chop it up more quickly. When we talk about fast oxidative fibers, they're still going to work for a long time because they're oxidative. We make a lot of ATP, but they will run out of ATP more quickly than a slow oxidative fiber would. So when we're talking about how quickly they'd run out of ATP, we might say eventually they run out of ATP. They fatigue, they're kind of moderate, if you will. So fatigue not fast or slow. I don't have a good way to describe that. Moderate fatigue rate. So it's going to take a little while for them to get fatigued, but they are going to fatigue more quickly than somebody that chops up their ATP slowly. When we talk about a fast glycolytic, the only way that these can make ATP is by chopping up the sugar that they already have stored. So I can't make any new ATP once I've used all of my stored sugar. So if I am a fast type of skeletal muscle and I can only break down sugar that I already have inside my muscle cell, these ones are going to run out of ATP really fast. So we would say that these run out of ATP really fast, or they fatigue quickly, because I have a limited amount of sugar and I'm chopping it up really quickly. Fast glycolytic fibers run out of ATP or they get tired really quickly. Slow oxidative fibers may not ever get tired, or they're going to get tired more slowly. The problem, though, with slow oxidative fibers is they give us a weaker muscle contraction because they're chopping up the ATP more slowly. So fast oxidative is kind of the best of both worlds. I'm oxidative, meaning I can make all kinds of ATP, and I'm fast, meaning I can chop it up quickly and make a really strong contraction. The one last thing for us to know about these types of muscle tissue is whether or not they need oxygen. And this one should be super easy based on the names of these types of muscle tissue. What word in the name of my types of muscle tissue tells me that they do need oxygen? What word in their name tells me we do need oxygen? Yeah, so it's not a trick question, right? That word oxidative means that I do need oxygen. I do need oxygen if I'm oxidative. If I'm glycolytic, I do not need oxygen. Oxidative, remember, meant that I use the electron transport chain to make my ATP, and at the end of the electron transport chain, I need oxygen. So these ones can't function without oxygen. This one can. Fast glycolytic can, because it doesn't take any oxygen for us to do glycolysis. We can just break stuff down. Look at my chat here. Yeah, so the question relates to those activities that are in your lesson packet to help you with pairing things up. When we're trying to figure out what types of muscle I would use for each activity, the big things that's going to help you make those predictions is how quickly they run out of ATP and how strong of a contraction. So the easiest one for us to make sense of, the kind of thing that slow oxidative muscle helps us with, this is going to help us with things like running a marathon. You don't have to have a particularly high amount of strength in your muscle contractions when you're running a marathon. It's the whole idea, slow and steady wins the race. So if you're running a marathon, we need to fatigue slowly. We just got to keep going. Slow oxidative, really helpful for that. When we're talking about something like a deadlift, where for just a few seconds you need to have a ton of energy, that's something that a fast glycolytic fiber would be helpful for. We make a really strong contraction. This is the strongest kind. Really strong contraction, but we can't do it for very long because we're just using what's stored. Fast glycolytic fibers, really good for a strong, short contraction. If I need a strong contraction, but I need to do it more than once or twice, like when you're talking about playing tennis, where that ball keeps coming back over the net. If I'm going to have to keep doing a muscle contraction, I need to keep making ATP. So a fast oxidative muscle fiber would be better for something like tennis. A deadlift, Miriam, is where you have just a really fast muscle contraction that you only have to do like once. Excuse me. If you look at the picture in your packet, it shows an Olympian with that really big set of barbells over his head. All he has to do is lift that one time and put it back down, and he's done. So that would be something that's called a deadlift, where you just do it really fast once, and then you're good. Go for it, Gloria.
Okay, so real quick, I know that in the tests and stuff, it was asking about myoglobin.
Uh-huh.
And so I know that myoglobin has a lot of oxygen, is that right?
Yeah, myoglobin transports oxygen. Yep.
So myoglobin is high in fast oxidative and slow oxidative, is that correct?
Correct. Yep, because those are the ones that need to have oxygen. So what Gloria is talking about here is, let's put lots of myoglobin. And we'll put it over here as well. Lots of myoglobin. This is like the younger brother of hemoglobin. So it's an oxygen transport protein that we find inside some of our muscle fibers, particularly inside the ones that use oxygen to build their ATP. So we'd see a lot of myoglobin in these two, my two types of oxidative muscle fibers. Not really going to see a lot of myoglobin in my glycolytic fibers because they don't need to transport oxygen like my oxidative ones do.
Okay, perfect. Thank you.
Mm-hmm. Any other muscle questions? Miriam, you had said you had a skin question. I don't know if you want to type that or say that. We can wrap up with that question.
Yes. So for the integumentary system, it's just knowing the or not the difference, but like when it asks for, when I was doing the exam review, when it says for the keratinocytes or creatine and melanocytes and everything like that, just knowing which one which one is involved, like if it's the epidermis or the dermis, like, because sometimes those throw me off. It's and it asks about what does the waterproof and things like that, and the glycolipids. So things that are associated with that just throw me off. It's if it's with the epidermis or if it's with the dermis.
Okay, so let me put that question to the class here. When we are talking about things like melanocytes and keratinocytes and those glycolipids, which part of the skin do we find those things in? Are those epidermis things or dermis things? Where do we find those things?
I know it's the epidermis for melanin.
Yep, so we find melanocytes and melanin in the epidermis.
Yeah. And I say I put epidermis as well for the keratinocytes, but that one I wasn't so sure of.
Yeah, so the entire epidermis is made out of keratinocytes. That's the name of the types of cells that are out there. They get their name because they're completely filled with keratin. So the epidermis made completely out of keratinocytes. Actually, Miriam, melanin is found in the epidermis as well. It just comes from the very bottom of the epidermis, that layer called stratum basal. So the melanocytes live in stratum basal. Keratinocytes live everywhere. All of those molecules that you mentioned, though, all of those are things that are found in the epidermis.
Because this was the question that was on the exam review that I was doing. It was saying it had like the blank and it said it's found with melanocytes and keratinocytes, and I just I selected the epidermis, but I wasn't so sure about keratinocytes. I was more sure about that it have melanocytes to prevent from UV radiation, because that's what the epidermis would do. But I wasn't so sure about the keratinocytes.
Yeah, so that's the kind of cell that makes up the epidermis. Yeah.
Okay. So keratinocytes makes up the epidermis, but melanin, it's found in the lower part of the dermis, correct?
It's the lower part of the epidermis.
Okay.
So if you look back, and we talked about it a little bit this morning, but if we look back, let me pull up a picture here. When you look at, bear with me, right here, when we look at a cross section of the epidermis, so all of this is epidermis cells. Most of these cells are keratinocytes, but this one right here that has the dark color to it, this would be a melanocyte. So there are very few melanocytes. They live in between a whole lot of keratinocytes that fill up the epidermis. But all of these types of cells that we're talking about, the keratinocytes and the melanocytes, they're both found in the epidermis layer.
Okay, got you. Yeah, that was the main question.
Okay, perfect.
Okay, and another one regarding the glycolipids and what makes it, and something about waterproof. It says waterproof and glycolipids. That one threw me off. It was saying either if it's the epidermis or if it's the dermis.
So do you know what makes glycolipids?
No, not really.
So glycolipids are so they're made out of sugar and fat. It's these little dots that you see inside my cells here. This row of cells is called stratum granulosum. So the glycolipids, also called lamellar granules, they're a special sugar fat, is what their name means, that's made by these keratinocyte cells that they spit out to waterproof them. So when we talk about glycolipids made by the keratinocytes, which layer of the skin would those be found in?
Epidermis?
In the epidermis as well. Yep.
Oh, okay. I put that as the dermis, but then again, I wasn't sure. I just left those questions off to ask later when we come in here.
Yeah, so those three things are found in the epidermis.
And I'm sorry, last question regarding the integumentary system is when it asks for the stages of melanoma, so it said which one, if it's also epidermis or dermis that's impacted by the stage 0 melanoma, and then it had which one is impacted by the stage 1 through 4 melanomas, and I put epidermis for the stage 0 and then dermis for stages 1 through 4, but I'm not sure.
When we talk about stage 1, we now I'm pausing. I want to make sure I tell you correctly. So stage 0 is confined within the epidermis, and after that point, as we start going deeper, we've gone into the very top part of the dermis. That's correct. I'll mention though, I'm not on the final exam. I'm not going to ask you about the skin cancer types. So it's mixed in because of the way that the questions were formatted, but that's not something to spend too much time on because I'm not going to ask you about that on the final.
Okay, I literally spent like I think 40 minutes trying to know the difference.
Yeah, so check out that learning objectives list to help you know where to spend your time. Yeah.
Gotcha. Okay, thank you so much.
All right, well, we are pretty much out of time for the night. I will be back. I mentioned kind of at the beginning of our time together that tomorrow morning at 9:30, we're doing the Salty Banana Challenge. So if you happen to have a banana and a little bit of salt around, you can come take a bite with us together tomorrow morning at 9:30. But I will stick around tomorrow from 9:30 to 11 to answer any last minute questions that you have. So if you're studying tonight and you come up with some more questions that you have, please feel free to email them to me so I can prepare to talk about them tomorrow morning. So last review session I will do with you is going to be tomorrow morning at 9:30. Unfortunately, Miriam, I will not be available tomorrow at 5, so it'll just be tomorrow morning. And if that time doesn't work for you, just shoot me an email, and I can try to reply via email too. All right, well, I'm going to stop the recording. To all of my friends planning to take the exam tonight, good luck. Get it done. You'll feel great when it's over. And for my friends planning to come back tomorrow, bring your banana, bring your questions. I will see you then.